Welcome to another episode of the MEGIN Spotlight series. Today we are delighted to have as our guest Professor Krish Singh from Cardiff University. Professor Singh is the head of Human Electrophysiology at Cardiff University Brain Research Imaging Centre, also known as Kubrick. Professor Singh's group uses MRI, EEG and MEG and has collaborated on research focusing on epilepsy, schizophrenia and Alzheimer's disease. Hi Professor Singh, thank you for joining us today.
Hi, morning.
So as an expert in electrophysiology, can you tell our audience what started your interest in MEG and how has its utilization evolved in your career?
Yeah, sure. So originally I'm from a physics and engineering background. As a kid, I was always interested in electronics and computing and building things, breaking things, programming things. This was back in the early days of computers, so you had to build anything that you wanted to use yourself. So I chose to do a physics degree. And during my physics degree, I learned about superconducting electronics and squids. And I also learned that you could use these technologies to study the brain, which I thought was pretty cool. So my PhD with Steve Swithenby at the Open University in the UK was about building squid detectors. So we used to build and design and build our own gradiometers and squids, which I really enjoyed. I wasn't particularly good at it, it has to be said. It was tricky technologically. And so I gradually came to realize that what I was really interested in was the sort of application side, the neuroscience questions that you can answer with this type of technology. So I was lucky enough to do a postdoc at Aston University, which at the time was really starting to get into MEG. And I was really lucky to go there right at the beginning of Aston's experiments with sort of multi-channel MEG. And that's where I met some of the people who are now long-term collaborators of myself. So people like Gareth Barnes, who's now head of the lab at UCL, Paul Furlong, who became the head of the lab at Aston, Ariane Hillebrand, who's now leading the MEG lab in Amsterdam, and many other people, Caroline Witten. You know, you can name many of the UK and European sort of leading MEG lab researchers, and all of us at one time or another had some kind of association with Aston. And that's really where my interest in using MEG to study brain function with a particular focus on the visual system really took off. And since then, I've kind of hidden away as a physicist in psychology departments. I'm currently in a psychology school at Cardiff. And I'm sort of really still excited by the range of applications that you can use with MEG to study the brain, both in sort of in terms of healthy brain function, but also in many different clinical conditions.
So I was lucky enough to visit you at the Kubrick Centre. So it's a very impressive facility and equipped with a great variety of modalities to support your research work. Can you tell us about the research focus and aims of Kubrick and why the centre believes MEG is so vital as a neuroimaging modality, and how does it support your research focus?
Yeah, so Kubrick's sort of overarching mission statement is to use multiple imaging technologies to get the most complete picture that you can of brain structure and function. So I'm sure you're aware that there's no one imaging technique that can do everything in terms of telling us how the brain works and what goes wrong in disease. And so you need multiple scanning technologies. And here in Kubrick, we have 4 MR scanners, in terms of variety of field strengths from 3 Tesla to 7 Tesla, and a high gradient connecton scanner, which is possibly the best technology we currently have for imaging the connectivity, white matter connectivity of the brain. And MEG and EEG obviously slot in terms of giving you a window onto the underlying electrophysiology of the brain. So looking at the sort of coordinated and coherent firing of neuronal populations and how that depends on synaptic modulations. MEG is, in my opinion, is really the only technique that we have that gives you a good non-invasive handle on that kind of signal. And so here in Qubit, we use MEG to do that, but then we also compare and contrast with other imaging techniques such as fMRI with its blood oxygen dependent response, which has this amazing spatial resolution throughout the brain down to sort of sub-millimeter, particularly at 7T. But the signals, the functional signals that you measure with fMRI are a hemodynamic signal that is only really related to underlying neuronal firing in a really complicated way that's difficult sometimes to understand. Whereas MEG, although we have some difficulties with spatial reconstruction that anybody who works with MEG will know about, we do have that sort of direct link right down to the sort of synaptic physiology. So MEG really fills that hole, that niche of being able to give you a true picture of what's happening in terms of electrical activity with reasonable spatial resolution.
So specifically on recent upcoming studies you've been involved in, where was MEG utilized and how was the data retrieved from MEG technology and how has it helped in the research studies and the findings?
Yeah, that's a good question. I think probably the best way to illustrate that is to talk about the two sort of major multimodal projects that we're currently running with MEG in Kubrick. They both the projects kind of illustrate the sort of dual focus that we have here. And the first focus that we have is on using MEG to provide essentially oscillatory fingerprints of what's going on in the brain in particular conditions. And because of the strength of the medical school here in Cardiff, one of the areas that we've recently been focusing on is psychosis and specifically schizophrenia. And the study that we're currently running with colleagues next door, which is called Converge, is a study of young people who have rare genetic variants, particularly deletions and duplications, which lead to a range of challenges for these young people. But one of them is that they have a very much increased potential risk of developing psychosis like schizophrenia. And this means that they are a very interesting group of people to study. So in Converge, we're going to be scanning around 120 of these young people and looking for signals within the brain using predominantly MEG actually, of where we can see oscillatory modulations in different frequency bands, which could tell us a little bit about what is happening in the brain right down to the synaptic level. And so I think this is a particular strength of MEG, that it gives you this window onto the sort of underlying neurobiology of some of these conditions like schizophrenia or genetic risk of schizophrenia that you just can't do with any other technique. So that's one area of focus. And I think, many groups are seeing MEG as a really powerful tool for sort of probing these sort of oscillatory fingerprints of conditions like schizophrenia and autism. And of course, we know about work that people are doing in epilepsy. The second sort of major project that we're doing that we're quite excited about is we're involved in a phase 1 clinical trial of a new drug, which is targeted at some of the cognitive symptoms of schizophrenia. It's a drug that initially was developed by colleagues here at Cardiff University in our Medicines Discovery Institute. And the drug is moving into its sort of first phase 1 clinical trial, safety testing and doing its initial first in human studies. And rather unusually, we've designed a MEG and fMRI study into that initial protocol, so that we're hopeful that we can show for the first time during this trial that there is a direct target engagement into the brain of the drug, and that we have some predictions based on the mechanistic action of this drug of what it should do to particular synaptic populations and connections, and how it will modulate the balance between excitation and inhibition. And so we can look for modulations of sort of gamma and alpha beta oscillations that we think will be sensitive to that modulation. And so that's part of our wider focus on promoting and using MEG for pharmacological applications, particularly in terms of helping accelerate the sort of drug discovery pipeline.
So could you tell us a little bit more about how the pharmacology connection came about with Cardiff University and how that connection has developed over time now?
Yeah, sure, of course. Yeah, happy to. I mean, we have a long-standing research program in the MEG lab here at Cardiff in using MEG to look at the pharmacological action of drugs. This was sort of mostly initially developed by my colleague Suresh Muthukumaraswamy, who's now leading an EEG pharma lab in New Zealand. And together with Suresh, or led by Suresh, we did many different studies, some involving psychedelic drugs like psilocybin, ketamine, and also anti-epileptic drugs like tiagabine. And our main interest there was trying to see whether MEG was a sensitive measure of the action of these drugs, and whether you could see unique signatures of action of each of the drugs that, in terms of the oscillatory profiles that I mentioned earlier. So, for example, when you give people ketamine has a profound effect across the brain on multiple frequency bands. And you can, with MEG, you have this unique opportunity to properly source localize those and map the effects of the drug across the brain and provide a kind of, as I said before, a fingerprint of where things are happening and what frequency bands are affected. And what's nice about that is that you can model the effects right down at the level of the synapse. So you can build little synaptic models in silico, which allow you to see which connections, for example, between pyramidal cells and inhibitory interneurons are being affected by the drug. And you do that by fitting these models to the measured MEG data. So MEG really gives you a deep insight into how these drugs are affecting the brain. So we were primarily, using pharmacomeg to try and develop and understand that linkage. But of course, it does offer up really exciting opportunities for helping drug companies that are bringing new drugs to the market to see whether their drugs are getting across the blood-brain barrier and having an effect on neurons, and also to understand whether the target engagement that you're seeing is matching the mechanism of action of the drug that's been designed. And that would be enormously beneficial to a drug company, because the drug development pipeline is really expensive, and anything you can do early on to inform the company's understanding of whether the compound is really doing what it should in humans is worth a lot to those companies. Now, there's been a lot of recent work in pharma using fMRI as a technique of the first choice, if you like, for doing these target engagement studies. And we do a lot of work using that ourselves with fMRI. We have colleagues here who've got long research programs of using fMRI to study drug action. But I guess what and many MEG colleagues across the world are starting to realize and trying to promote is the idea that MEG is, in a way, a better technique for doing these types of studies, because of its direct link to the underlying neural activity. It doesn't have this problem that you're seeing function through this kind of complicated and cloudy hemodynamic lens that the fMRI signal does. So we think MEG and its ability to spatially localize these direct electrical effects provides a really exciting new tool for pharma to help them accelerate and improve the drug development pipeline for novel compounds. So that's why we are excited about it here in Cardiff, and why we're spending quite a lot of time developing and focusing on this as one of our core areas of activity.
So do you see then the MEG and pharma work as translational research, I guess, and in the future is the aim that Kubrick's current research will support new developments and clinical applications for MEG this way?
Well, I think it's translational in the sense that we're helping pharma companies to evaluate and bring new compounds and drugs to the market. So in a way, it is bringing us closer to patient treatment than we would be if we weren't involved in this space. Yeah.
You're also very involved with MEG UK, and for anyone in our audience not already involved or familiar with this group, can you share a little bit about the aims of the group and how they can participate? I was recently in Germany, actually at a symposium, and it was a renowned group, so everyone was talking about it. So it might be worthwhile mentioning it.
I think this, out of many things in my career, this is one of the things that I'm most proud of and pleased about is the way that the UK MEG community has been so well linked up and collaborative. I mentioned that I was at Aston. First off, I was there in the early 90s and then again in the early 2000s. And many people who are involved in UK MEG research started at Aston or had some association at some time. And it meant that I think there was a really nice community focus that came from Aston originally. And it meant that as people moved around the UK and started opening their own MEG labs, we could maintain that connection. So for example, Matt Brooks, who's head of the MEG lab at Nottingham, he spent quite a lot of time during his PhD at Aston before he moved back and helped develop the MEG lab at Nottingham. So we had this really great link of people who wanted to work together and wanted to drive MEG forward, because MEG was a very embryonic technique in the UK. We all felt that we needed to build it. We were all passionate and evangelical about MEG. And it meant that we kept having these yearly meetings. We kept sharing advice and collaborating on papers. And, even to this day, we've managed to maintain that grouping. And we were very lucky because we were actually funded by the MRC, which is one of the main UK research funders for clinical research, in around 2011, 2012, I think, that we were given a grant, which was roughly about £1 million in total, to really link up all the MEG labs in the UK and drive collaborative activity, have a small cohort of PhD students working across the sites, build an embryonic shared database and collect some multi-site data, and also importantly fund meetings every year, which we called MEG UK. And I think because of that, we've really helped. That's really helped maintain this collaborative grouping, which we call MEG UK, and we now call MEG UK I. Importantly, we've added the Dublin site in Ireland, so we're now MEG UK I. And I think across the world, lots of national groupings or regional groupings have seen what we did and have started to create similar collaborative infrastructure. So the one that I'm most familiar with is MEG Nord, which I think is also quite successful now. What I should say is that our annual meeting, MEG UK and now MEG UK I, we used to get 50 to 100 people. We now comfortably get 200, 250 people each year coming to the meeting. And a lot of people come from the rest of Europe to these meetings. We're very open. Anybody can come. It's not, you don't have to be working in the UK. And I think that sort of open and welcoming attitude has stood us in good stead, and long may it continue.
Well, I think you certainly made an impression, like I said recently, in meeting a group of MEG scientists in Germany. They would like to emulate, I think, a similar group for Germany. So I think you've made a good impression and have a good reputation for people to follow.
Yes, yeah, I was just one of the members. It wasn't my, I should say, when it was a group effort by all. Well, originally it was 8 MEG labs. Now I think we're up to 11 or 12.
Wow, that's great.
Kind of lost track now. Yeah. But it's great. And as I said at the beginning of my answer, it's something that I'm sort of really pleased and excited about. Yeah,
no, you should be. It seems like it's been a great success. So thank you. Thank you very much. We've come to the end of our session today. So thank you very much for joining us today. And it's been incredibly insightful of what the work you're doing at Kubrick, and appreciate your time today. Thank you.
Thank you. My pleasure.